Hello and welcome to our group's video presentation for Biology SEL. The topic our group will be presenting is Chapter 42.1, Early Developmental Stages. Before we start, let us introduce ourselves.
Hi, my name is Aida Binti Mohamad Shah, and I will present about the cellular stage of embryonic development.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nabil bin Mahadzir, and I am from Group Lecture 6. Today, I am going to present about the organ stages of development.
Bismillahirrahmanirrahim. Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is Nurain Afina Binti Abbas, AS10466. In my part of this presentation, I will explain to you about the tissue stages of development.
Hi, my name is Ying Elaine, and I will talk about the details of fertilization in humans.
Before we start with our presentation about early development stages, there are a few learning outcomes that we should know. Firstly, identify the structure of an egg and sperm that are directly involved in fertilization. Second, compare and contrast cellular, tissue, organ stages of embryonic development. And lastly, describe the major steps in the development of central nervous system of vertebrates. These are the things that we should know by the end of this presentation.
Okay, let's understand the details of fertilization in humans before moving on to embryonic development. As we all know, fertilization is the union of a sperm and an egg to form a zygote. And from that we know that the sperm and the egg play an important role in fertilization in humans. So first, let's take a look at the human sperm. The human sperm has three distinct parts: the tail, the middle piece, and the head. The head contains the sperm's nucleus and is capped by a membrane-bound acrosome. The acrosome secretes digestive enzymes to forge a pathway for the sperm through the zona pellucida, which I will explain later. Next, let's take a look at the egg of a mammal, which is actually the secondary oocyte. Now, the secondary oocyte is surrounded by a few layers of follicular cells. Collectively, they're called the corona radiata. These follicular cells nourishes the oocyte when it is still in a follicle of the ovary. Next is the zona pellucida, which I mentioned earlier. It is an extracellular matrix just outside the plasma membrane of the oocyte, but beneath the corona radiata. Okay, so now that I've introduced the parts of the human sperm and the human egg, let's start to understand the process of fertilization. Firstly, millions of sperm begin their journey to fertilize the oocyte, but in the end, only one sperm actually fertilizes the oocyte. So the sperm basically covers the surface of the oocyte and secrete enzymes to weaken the corona radiata. Then they try to squeeze through the corona radiata and bind to the zona pellucida. So when a sperm's head manages to bind tightly to the zona pellucida, the acrosome releases digestive enzymes to forge a pathway through the zona pellucida, as I mentioned earlier. So when the first sperm successfully binds to the plasma membrane, a few events will take place to prevent polyspermy. Now, polyspermy means the entrance of more than one sperm. So how this works is that as soon as the sperm touches the plasma membrane of the oocyte's plasma membrane immediately depolarizes, and this changes the charges of the plasma membrane. And this is known as fast block, as it repels other sperm from entering only for a few seconds. Then we have the cortical granules, which are basically vesicles in the oocyte that secrete enzymes to turn the zona pellucida into an impenetrable fertilization membrane. This is a longer lasting reaction, and it's called the slow block. During this phase, Juno molecules are also released from the oocyte. The Juno molecule is basically a key molecule present under the zona pellucida. It is a molecule that is required for sperm-egg fusion, and in this phase, it prevents further specific interactions. Now, after all that are the last few events of fertilization that includes the formation of diploid zygote. The plasma membrane of the oocyte actually has microvilli, which extends from the membrane, and its function is to bring the entire sperm into the oocyte. Now, as the sperm has entered the oocyte, the sperm's nucleus releases its chromatin, which will reform into chromosomes enclosed within the sperm pronucleus. While that is happening, the secondary oocyte completes meiosis and becomes an egg where the chromosomes are also enclosed in a pronucleus. Then, both sperm and egg pronuclei are surrounded by a single nuclear envelope. From there, cell division takes place, and also the basal body of the sperm's flagellum is where the centrosomes that will give rise to a spindle apparatus are derived from. And the two haploid sets of chromosomes will share the first spindle apparatus of the newly formed zygote. Next, we will talk about embryonic development, which I will pass to Aida.
Okay, let's move on to embryonic development. Embryonic development are divided into three parts: cellular stage, tissue stage, and organ stage. At cellular stage, we will learn about cleavage and formation of blastula, while gastrulation we will know at the tissue stage. This is the figure of embryonic stage of development of chordates, including human. From this figure, we can divide it into two parts. At the left side is the cleavage, while the right side, the blue, is where the gastrulation occurs. For my part, I will explain about cellular stages of development. Okay, before I explain further about the flow, what is cleavage? Cleavage is cell division without growth, where DNA replication and mitotic cell division occur repeatedly. We can also describe cleavage as cell division without cytoplasmic addition and enlargement. So, as a result, the cells get smaller with each division. Basically, cleavage occurs during the first stage of animal development. Okay, let's start with the cellular stage. It will start with zygote, morula, and blastula. Shortly after fertilization, zygote undergo a series of rapid mitotic cell division. We call it as cleavage. It initially gives rise to the morula, which is basically a solid ball of unspecialized cells. As cleavage continues, the cell of the morula pumps sodium ions into extracellular spaces. As this occurs, water follows by osmosis. The water collects in the center, so it forms blastula. Blastula is a hollow ball of cells. The fluid-filled area in the center is called a blastocele. Once the blastocele is formed, the next stage of embryonic development, called gastrulation, begins. Okay, next, there are different appearances of blastula. As I explained earlier, that's for the lanceolate, but it will have a different appearance for the chicken and frog. As you all know, chicken lays a hard-shell egg containing yolk. Because of the yolk, cells do not participate in cleavage. For chicken, the blastula is a layer of cells that spreads out over yolk. Based on the figure, blastoceles that forms as a space that separates these cells from the yolk. Okay, next, in frog, cleavage is also not equal because of the presence of yolk. So when there is yolk, zygote and embryo exhibits polarity, and the embryo has an animal pole and vegetal pole. Actually, animal pole contain faster growing, smaller cells that will develop into ectoderm and endoderm, while vegetal pole contains slower growing and larger cells that develop into endoderm. Ectoderm and endoderm will be explained later. Actually, the functions of blastocele in frog embryos are: it permits cell migration during gastrulation, and second, it prevents the cell beneath it from interacting prematurely with the cells above it. Okay, that's all for my part. I will pass to Afina for the tissue stages of development.
Basically, tissue stages of development involve gastrulation. Gastrulation means the formation of gastrula. So what is gastrula? Gastrula is an embryo at the stage following the blastula when it is a hollow cup-shaped structure having 3 layers of cells. During gastrulation, the blastula made up of one layer falls inward and enlarges to create the 3 layers of cells. Each of these layers is called a germ layer, and each germ layer differentiates into different organ systems. It means that gastrula is known as a 3-layered structure. These 3 germ layers are known as the ectoderm, mesoderm, and endoderm. Okay, gastrulation is divided into 2, which are early gastrula and late gastrula. An early gastrula has 2 layers of cell, including the ectoderm and endoderm. The ectoderm is the outer layer of cells, while the endoderm is the inner layer. In addition to ectoderm and endoderm, the late gastrula has a middle layer of cells called the mesoderm. Now, I will explain about the diagram here. First of all, from the blastula, the vegetal pole of the blastula flattens, and then some cells change shape and move inward to form the acantheron. The opening into the acantheron is the blastopore. This is when the gastrulation occurs, and then the early gastrula started to form. The outer layer of cells is now ectoderm, and the wall of the acantheron is endoderm and future mesoderm. And finally, the late gastrula form, having a middle layer of cells called the mesoderm. Let's proceed to the next part. I will explain to you about the development of mesoderm. In some animal, how the mesoderm develop is all different. Now, I will compare the development of mesoderm between 3 animals, which are the lancelet, frog, and chick. In lancelet, mesoderm forms by outpocating of the acantheron. In frog, mesoderm forms by migration of cells between the ectoderm and endoderm, while in chick, mesoderm forms by invasion of cells. To be more detailed, in lancelet, these outpocatings grow in size until they meet and fuse, forming 2 layers of mesoderm. The space between them is the coelom. The coelom is a body cavity lined by mesoderm that contains internal organs. Next, in the frog, cells containing yolk do not participate in gastrulation, and therefore they do not invaginate. Instead, a slit-like blastopore is formed when the animal pole cells begin to invaginate from above, forming endoderm. Animal pole cells also move down over the yolk to invaginate from below. Some yolk cells, which remain temporarily in the region of the blastopore, are called the yolk plug. Mesoderm forms when cells migrate between the ectoderm and endoderm. Later, the mesoderm splits, creating the coelom. For chick, an upper layer of cells become ectoderm, and a lower layer becomes endoderm. Mesoderm arise by an invagination of cells along the edges of a longitudinal furrow in the midline of the embryo. Because of its appearance, this furrow is called the primitive streak. Later, the newly formed mesoderm splits to produce a coelomic cavity. In conclusion, no matter how gastrulation takes place, the result is always the same. 3 germ layers will be formed. That's all from me. Thank you. Assalamualaikum.
Hello, so I'm going to explain about the organ stages of development. So you can refer to the graph or image that I have attached on the slide. So we can see that the newly formed mesoderm cells that lie along the main longitudinal axis of the animal and coalesce to form a dorsal supporting rod called the notochord. Organ formation can be related to germ layers. For example, the nervous system developed from midline ectoderm. The ectoderm is the outer layer and it is located just above the notochord. During this process, a layer of cells called the neural plate contributes to the formation of a neural tube. The anterior part will develop into the brain and the rest will become the spinal cord. The neural crest is a band of cells that develops where the neural tube pinches off from the ectoderm. These cells migrate to various locations where they help from skin, muscles, the adrenal medulla and the ganglia of the peripheral nervous system. Midline mesoderm cells. Mesoderm, if you can see at the image, mesoderm is the one that at the middle of the thing. So these midline mesoderm cells that did not contribute to the formation of the notochord now become two longitudinal masses of tissue. These two masses become blocked off into somites. These somites it will eventually give rise to muscles associated with the exoskeleton and to the vertebrae. Lateral to the somites, the mesoderm will split, forming the mesodermal lining of the column. Okay, that's all. Thank you.